Hello class. Today we're going to try something a little different. We are going to be doing what is called a flipped lesson. So I gave you a handout at the end of class and this is what that was for. So I'm going to cover a topic and there are guided notes that I gave you and you're going to fill them out as I go. So you may remember from the last class or 2 classes ago that we were talking about mitosis. Well, now we are going to be covering meiosis, which is similar to that process. So you may be wondering what meiosis is. It's the form of cell division by which gametes with half the number of chromosomes are produced. So in mitosis, diploid cells were formed, but in meiosis, haploid cells are formed. And meiosis is asexual reproduction. And there are two divisions which are easy to remember by name, meiosis 1 and meiosis 2. A little bit more about meiosis. Sex cells divide to produce gametes, or sperm or egg. Gametes have half the number of chromosomes. And these occur in the gonads, or testes for the male, ovaries for the female. In males, they go through what is called spermatogenesis, which deals with the sperm, and females go through oogenesis, which deals with the egg. So first, spermatogenesis. Start with the human sex cell, 46 chromosomes. That breaks in half and forms two identical diploid cells, which then goes on to form 4 identical haploid cells. And then oogenesis basically starts the same way. You got 46 chromosomes, breaks apart in meiosis 1, forms 23, and 23, 2 identical, and then goes on to form 1 haploid cell and 3 polar bodies, and the polar bodies eventually end up dying off. So now you may remember from mitosis that there was interphase. Well, there's interphase in meiosis also. So this phase in meiosis is similar to that of mitosis, and chromosomes replicate in the S phase. Each duplicated chromosome consists of 2 identical sister chromatids attached at their centromeres. Centriole pairs also replicate. So as you can see in this picture down here, you start with what looks like a half X. That is one part of it, and then it replicates and forms a double-stranded chromosome. A little bit more about interphase 1. The nucleus and nucleolus are visible. So as you can see, the arrows point those out. Surrounding these, you can see the nucleus, nucleolus, the chromatin, and the cell membrane protecting that cell. So now we move on to meiosis 1. And this is where cell division that reduces the chromosome number by 1/2. And as you may remember from mitosis, there was 4 phases in mitosis. There's also 4 phases in meiosis 1. And those are prophase 1, metaphase 1, anaphase 1, and telophase 1. All the same as mitosis, just with a 1 after it. So in prophase 1, this is the longest and most complex phase. This takes up 90% of the time of the cell dividing up, splitting apart. So during this phase, chromosomes condense, synapsis occurs, which is when homologous chromosomes come together to form a tetrad. And a tetrad is 2 chromosomes or 4 chromatids. And we are going to look at a picture now, and it might make it a little more clear to you. So basically, synapsis, you start with 1 on this side and 1 on the other side, and then they basically form an identical copy of themselves. So as you can see, you got 2 homologous chromosomes, sister chromatids, and they form what comes the tetrad. A little bit more on homologous chromosomes. They are a pair of chromosomes that are similar in shape and color. Homologous pairs, or tetrads, carry genes controlling the same inherited traits. Each position of a gene is in the same position on homologues. Humans have 23 pairs of homologous chromosomes. The first 22 pairs are autosomes, and the last pair is what considers the sex of baby. And this one kind of breaks it down a little bit. It talks about eye color and hair color, and it's just a little bit of the chromosome though. So it marked it out. It has the arrows pointing to where on the chromosome determines the eye color and where the hair color would be determined from. And then these small ones are male chromosomes and these big ones are female. So now we're going to discuss crossing over. Crossing over may occur between non-sister chromatids at sites called chiasmata. Crossing over is when segments of non-sister chromatids break and reattach to the other chromatid. Chiasmata are when chromosomes touch each other and exchange genes, and this causes genetic recombination. And there's a picture down there. You have two non-sister chromatids, and they cross over. As you can see, they're crossing over each other. And then eventually, you got one copy from one chromatid, one copy from the other, just attached on separate ones, on the separate tetrads. So a little bit more about synapsis. Synapsis is pairing of homologous chromosomes. So it's basically what occurs during crossing over. So you have a paternal side and a maternal side. The paternal side crosses over the maternal, and then as you can see, they start to change. The orange one gets the maroon side on it, and the maroon side gets orange. And then at the end, you got an orange one that's all orange except for one leg turned maroon, and the same on the other one. You got all maroon, and then one piece that is orange. Now we're going to discuss sex chromosomes. So if you are a female, you have 2X chromosome. That's what defines you as a female. And if you are a male, your XY chromosome. So as you can see, X chromosomes are the larger ones. That's why you have two right here. That's why it's a female. And then over here, you have one big one and one little one. The big one is an X, and the little one is a Y, and that's what makes a boy. Meiosis 1, the first phase, prophase. During prophase, the nucleus and nucleolus disappear, which you may remember they were visible in the stage before. The spindle forms, chromosomes coil, and synapsis occurs. Tetrads form, and crossing over occurs. So as you can see, spindles form. There's the tetrads. The second phase, metaphase 1. This is the shortest phase. Tetrads align in the middle of the chromosome. Independent assortment occurs. Chromosomes separate randomly, causing genetic recombination. So these images right here show the tetrads aligning in the middle of the cell on both of them. And then they have different gametes, which we discussed at the beginning of the slideshow. The next phase, anaphase 1, homologous chromosomes separate and move towards the poles, and sister chromatids remain attached at their centromeres. And this is a picture describing what is going on during that phase. In the last phase, telophase 1, each pole now has haploid set of chromosomes, and then cytokinesis occurs and two haploid daughter cells are formed. So as you can see, it looks like there's two cells forming right here. Eventually, during cytokinesis, they split apart, which we're going to talk about here after this. And they have identical tetrads. And there's another picture depicting what is going on. A little clearer, you can see more so that they are almost going to split in half and form two haploid cells. Meiosis II, there is no interphase during this, or it may be like a very short process, you won't even notice it. No DNA replication takes place, and meiosis II is very similar to mitosis. Prophase II, it's the same as prophase and mitosis. Nucleus and nucleolus disappear, chromosomes condense, spindle forms. So basically what we just talked about in prophase 1, that is the same thing that is occurring in prophase 2, but with two different cells instead of just one. Metaphase 2, same as metaphase and mitosis. Cells line up in the middle, as you can see. Basically the same as what we just talked about, like I said, but two chromosomes instead of one. Anaphase 2, same as anaphase and mitosis. Sister chromatids separate. So as you can see, chromatids separated in both of these. In telophase 2, same as telophase and mitosis. Nuclei and nucleoli reform. Spindle disappears. Cytokinesis occurs, and 4 haploid daughter cells are produced. So as you can see, just like we talked about in telophase 1, they're going to split, but this time it's with 2 of them. And then they form 4 haploid daughter cells. And that is the end of the slideshow. So if you need to go back to finish your guided notes, you are welcome to. And tomorrow we are going to be doing we're going to review over what this was about, and then we are going to be doing a little stations activity. That should be pretty fun for you guys. So thank you for watching, and I'll see you guys tomorrow.